We'll talk a little bit more about each one of those components a little bit later. So once we get past the stomach itself and we think about the glandular organs, we have other associated organs such as the pancreas and the liver. So you know from the stomach we're going to get gastric juices and acids and pepsin. From the pancreas we're going to get enzymes and buffers, and then from the liver we're going to get bile. When we look at the components of the intestine, you can see that we have the small intestine that's composed of the duodenum, the jejunum, and the ileum. And then we have the large intestine. Part of it's been removed right here. You can see it continues right here, right? When we take a section of the small intestine, such as this section from the jejunum, you can actually see the plica that we talked about earlier, those permanent fold. Right, so 90% of nutrient absorption occurs in the small intestine, and it is incredibly important in digestion and the absorption of nutrients. The first section is called the duodenum. It's about 10 inches long, and it is called the mixing bowl. And this is where I'd really like for you to think about what you might have considered to be the mixing bowl before. Most students typically say at this point that they consider the mixing bowl to be the stomach, and it's actually not. It is considered to be the duodenum, because not only do you have chyme from the stomach, which is all of the food that you ate that became a bolus, that became more and more digested, and then you have the secretions from the stomach. Those two combined make chyme. So chyme is what actually enters the small intestine. All right, so you have chyme in the mixing bowl, and then you have secretions from both the pancreas and the liver, and that is the reason why it's called the mixing bowl. The jejunum is the middle section. It's about 8.2 feet long. This is where most chemical digestion and nutrient absorption occurs, which makes sense. It's a very long portion. And then the ileum is the final segment. It's almost 12 feet in length. The plica that we talked about before are these transverse folds. They're permanent and they do not disappear when the intestine fills. So here we have the small intestine. You can see the permanent plica. And with the permanent plica, they have tiny little villi. So you can see here are the villi. And when we take a villi apart and look at it, you can see that there are lymph vessels, there are there's arterial blood, there's venous blood that you would find within a capillary. Also notice we have goblet cells, we have nerves in here. So these villi are these finger-like projections. They're in the mucosa and they are covered with the microvilli. And that is what you can see in this image. The intestinal glands are also found in here. They're between the columnar epithelial cells, you can see right here, and they're going to allow for mucus to be pushed onto the surface of the intestine. The duodenum is different in that they have fewer plica than the jejunum, and they have small villi. Because the function of the duodenum is to be that mixing bowl. And once you receive all of the substances from the stomach, you know that hydrochloric acid is in there. And so one of the functions of the duodenum is to neutralize acid before it gets to the absorptive surfaces of the small intestine. The intestinal secretions that we find is very watery. Almost 2 liters enter the lumen of the intestine, and it gets the chyme moistened, helps to buffer, and it keeps everything in solution. The intestines move. You have very weak peristaltic contractions, as in the duodenum, as you're trying to move the contents towards the jejunum. Associated with the small intestine, we have the organ that is called the pancreas. And the pancreas is really interesting. It's called a reticular organ. If you remember, I've said to you in the past that a reticular organ is like a very delicate bag that is filled with kernels or um, small individual pieces that come together to form the organ. Your liver is a reticular organ, and your spleen is a reticular organ, and your pancreas is a reticular organ. In this image, I want you to notice that we have, first let me erase this line I just drew. In this image, I want you to notice that we have a portion of the small intestine, and we also have the pancreas. But look here, the area that I've just circled, I want you to notice the common bile duct coming from the liver and the pancreatic duct that is part of the pancreas. And notice how they come together right here at the duodenal ampulla, right? Something to think about. So we have substances from the pancreas, substances from the liver, all come together in the mixing bowl that is the duodenum. As you can see, it's posterior to the stomach. It's actually bound to the back wall, posterior wall of your abdominal cavity, and it has this very thin connective tissue capsule. Remember, again, this is what makes it a reticular organ. The pancreas, much like many other organs, has the different regions, such as the head, the body, and the tail. So, a commemorative name for the pancreatic duct is called the duct of Horsig. And I've already highlighted the pancreatic duct for you. This is where you get the digestive enzymes and buffers that come from the pancreas and allows them to be dumped into the duodenum at the duodenal ampulla. The common bile duct goes from the liver and the gallbladder to the duodenum. It meets the pancreatic duct at the duodenal ampulla. This is the chamber that receives the secretions from both the pancreas and the liver. And interestingly, it is about halfway the length of the duodenum. It's not where it connects to the stomach. It's not where it connects to the jejunum. It's about halfway. This allows for the contents of the stomach to actually enter the small intestine and to be mixed before it gets to the jejunum. As I noted before, the pancreas has these small kernel-like structures. These are called lobules. They have blood vessels. Each one of the lobules branches, and they allow for different products to be produced. Okay? So the endocrine tissue is also part of the pancreas. So it's not just things that we need for digestion, but some have an endocrine activity. So we have endocrine cells that secrete insulin and glucagon into the bloodstream. And then we have exocrine cells, and they are the epithelial cells of the duct system. Your pancreas secretes about 1,000 milliliters, or about a quart, of pancreatic juice per day. Your duodenum is in control of what your pancreas does. That's because of the hormones that come from the duodenum. And these pancreatic secretions contain pancreatic enzymes. Pancreatic enzymes include a carbohydrate, alpha amylase, a lipase, pancreatic lipase, that breaks down complex lipids, and then it has nucleases. So and one more, we also have the enzymes that break down proteins. So if you look at your pancreas, you are breaking down carbohydrates, you're breaking down lipids, you're breaking down nucleases, and you are breaking down proteins. So when we think about the large organics, we've captured them all here, and the pancreas helps with digesting all of them. So if you look at the proteolytic enzymes, those that break down proteins, we have proteases that break down large complex proteins and peptidases that break down the smaller peptides into amino acids. The proteolytic enzymes, it's 70% of everything that is produced in regard to enzymes in your pancreas. They only become activated once they reach the small intestine at the level of the duodenum through the duodenum. So your liver is another accessory organ. It is the largest visceral organ that you have. It's in the right hypochondric region and epigastric region. So if you remember, it is because of your liver that your left lung is slightly elevated. And because of your liver, you have metabolic and synthetic activities. So just looking very quickly at the anatomy of your liver, you have the right lobe and the left lobe. Right lobe is much bigger. We're looking at the anterior surface, so right lobe, left lobe. If you flip it around, look at the posterior surface. Your left lobe, as you can see, is quite a bit smaller. Here is your right lobe, but also you can see this tiny little lobe right here that is called your caudate lobe. Also at the posterior surface, you can see your gallbladder, the full gallbladder. It's about the size of your thumb. If you go back over here and look at the interior surface, you can see where it peaks out between your stomach and your liver. The falciform ligament is found on the anterior surface, and it divides the right and the left lobe. Right? Here's your falciform ligament. On the posterior surface, again, note the small caudate lobe. The quadrate lobe is inferior to the caudate lobe. There it is. There's the little guy. And it's between the left lobe and the gallbladder. So when we think about the liver, we need to think about bile. It has an incredible duct system. There are narrow channels, and you remember that narrow channels are also called canaliculi. And these bile canaliculi extend outward from the central vein, and they connect with the ductules that actually carry bile to the bile ducts. So your right and left hepatic duct. So if you see the word hepatic, you must think liver. It collects all the bile from the bile ducts in your liver, and they come together to form the common hepatic duct, which leaves the liver. The common hepatic duct will either go to the common bile duct, which empties into the duodenal ampulla, or to the cystic duct, which leads to the gallbladder. So from the common bile duct, you can either send the bile one of two ways, to the common bile duct or to the cystic duct, and that will lead to the gallbladder. The common bile duct, again, is formed by the union of the cystic duct and the common hepatic duct. It passes through the lesser omentum toward the stomach. It then penetrates the wall of your small intestine, and it meets the pancreatic duct at the ampulla. So what does your liver do? Helps with metabolism. It helps with blood regulation or hematological regulation, and it also helps with bile production. So if you look at the metabolic regulation, that's going to help with the composition of circulating blood, how you metabolize nutrients, how you remove waste, the storage of nutrients, and drug inactivation. With the hematological activities, everything that leaves the absorptive surfaces of your digestive tract is going to flow into the liver. So once it's in the liver, nutrients are extracted or toxins are extracted, and before it actually gets to the systemic circulation. Excess nutrients are going to be removed and stored in your liver, and it actually has the ability to correct deficiencies. So you can either keep nutrients in storage or you can mobilize them so that they can go out into the system and be utilized where they're needed. And you actually can have activities, synthetic activities that occur in your liver as well. So when you think about how important your liver is, you metabolize carbs, lipids, amino acids. All of this occurs in the liver. So if you think about someone who has liver disease, what can be impacted? Waste product removal. This is a huge one. Storage of vitamins, mineral storage, and again, drug inactivation. So this is the largest blood reservoir in your body. You have 25% of cardiac output is in your liver at any one time. And the 6 blood regulation functions are as follows. So phagocytosis and antigen presentation in your blood occur because of the activities in your liver. The making of plasma proteins such as albumin. The removal of circulating hormones that are no longer necessary. Let's say you produce a hormone, negative feedback loop tells your body that you've produced enough. Well, then those circulating hormones will be removed. Antibodies are removed as necessary. Toxins can be removed or stored. And then you also make the synthesis or make bile and secrete bile. So let's go to the removal and storage of toxins. If we were in class, I would get up on my soapbox and say to you, be careful about eating liver. For every mammal, it is a filter to remove toxins and to remove waste. So we have to really think about, is this something that is appropriate to be eaten? Do you know where the liver that you're eating came from? Do you know what the animal was exposed to? So also when we think of your liver and you think of your gallbladder, we have to think about lipids. When you ingest lipids, we know that they're just based on what a lipid is, it's not water soluble. The processing of lipids in your stomach the mechanical processing allows for the lipids to be broken down into large drops. Once you get the lipids into your small intestine, the pancreatic lipase isn't lipid soluble, so it can only interact at the surface of the droplets. So think about, salad dressing that you have to shake up, where you have the olive oil that has floated to the top, and you shake it a little bit, and you have some larger droplets, but they're not completely, the salad dressing is not completely emulsified. Well, how do you get that to be completely emulsified? Well, it's because of bile in your body that allows for the droplets to be broken apart. Now, if you're shaking salad dressing, if you continue to shake it, and you continue and continue to shake it, you'll actually get to where you have just tiny droplets of oil. Well, that doesn't happen in your gut. So you have to have some additional assistance with this. And it's because of bile salts that you can have this emulsification where you're breaking up the big droplets to where they're smaller droplets. So if you think about where bile is stored, it's in your gallbladder. And this is the place where you keep these droplets that are going to help you break down fat. So let's just think for just a minute about what happens to people who have had their gallbladder removed. Well, for some, it's very difficult for them to eat fats after they have their gallbladder removed because their body cannot process the fat. And so they have fatty-filled, oily diarrhea, and it takes some time for their body to be able to process fats at all. And for most people that have had their gallbladder removed, they need to go on a low-fat diet. So the gallbladder, pear-shaped muscular sac, you know where it's located. It stores and concentrates bile prior to it being excreted into your small intestine. Again, it has three regions: the fundus, the body, and the neck. So you can see in this image where your gallbladder is located, right? But we have another structure that is called your cystic duct. I'm going to change colors to make it a little easier to see. This is your cystic duct. It forms a union with the common hepatic duct that you can see right here to form the common bile duct that I'm circling right here. The common bile duct, this structure right here, will meet the pancreatic duct, as you can see right here, before emptying into the small intestine at the duodenal end. So this ampulla receives substances from the pancreas and substances from the liver and the gallbladder, meaning bile. We've already talked about the fact that the gallbladder stores bile and it releases the bile into the duodenum. But here's where the hormones come in. They only do it under stimulation of the hormone cholecystokinin, shorthanded as CCK, right? So bile is released by the cues that come from the hormone cholecystokinin. So if you think about your gallbladder and how much it contains, 40 to 70 milliliters of bile, but as the bile sits in the gallbladder, the composition changes because concentration, it's not just storage, but concentration of the bile also occurs in the gallbladder. So water is absorbed and the bile salts and the solutes become concentrated. Well, you could very easily, if the concentration is too great, or you do not have enough water in your diet, or you have a fatty diet, you could get gallstones. Now, don't think that everybody that gets gallstones is not drinking enough water or has too fatty of a diet. Some people just have a greater propensity, familial propensity to get gallstones. So these are crystals of the bile material. They're insoluble. These are minerals and salts. If the bile is too concentrated, they will form scones. Before stones are formed, a person may actually form something that is like a paste. And the first indications that there are problems with their gallbladder is when they have slight pain on either the center of their chest or a little bit to the right. And that may be when this paste is trying to be passed by your body. Now, gallstones can plug up a gallbladder, and when that happens, the gallbladder can become inflamed, infected, and it can be very painful.